In this video, we're going to dive into protein structure. We talked about the amino acids individually, and now when those amino acids all come together, they're going to form the proteins. So amino acids are the building blocks. Proteins are the more functional units from those building blocks. Starting out, the characteristic bond of all of our proteins is known as the peptide bond. This is the base bond. It is a covalent type bond, and it is a carbon-nitrogen bond between the carboxyl group and the amino acid, or the amino group, specifically in each of the amino acids. So remember earlier we looked at what the actual amino acid looks like. It has a carboxyl group, a hydrogen, an R group, and then an amino group. So that carboxyl group and the amino group form a bond in order to, the carboxyl group of one amino acid forms a bond with the amino group of another amino acid to form a long chain. This is our primary structure. It is also called, taking one step back, our peptide bonds are also known as covalent. They're also known as amide bonds. Okay, these are all AKAs of each other. You can see any of these, they all will mean the same thing. They're all perfectly good explanations as far as perfectly good answer choices for what is the characteristic bond of all of our proteins. The primary structure of proteins is the linear sequence of amino acids. So we join all of the amino acids together and we form this chain. That is our primary structure. It has the peptide bonds. We make a polypeptide chain. Poly means multiple. Peptide referring to those peptide bonds, the amino acids. Chain meaning they're all linked together. Our secondary structure, once we have our chain formed, remember how we have all of those different R groups, which give each of our amino acids slightly different properties, different chemical properties. When these are all right next to each other, they're all going to interact with one another chemically, okay? And they're going to fold into either alpha helices and or beta pleated sheets. So you can have multiple alpha helices and multiple beta pleated sheets within a single protein. It just depends on how all of the different amino acids interact with each other. And they're interacting with each other through their hydrogen. So you get these hydrogen bonds. So everything folds. The way that things are attracted or repelled from each other, we know that, opposites attract. Positive charge goes towards negative charge. Negative charge repel each other. It's basically what happens is everything shifts and moves in order to where it's attracted and where it's repelled in order to create these folds. Alpha helices, beta pleated sheets. We can link those to our secondary structure. Primarily, hydrogen bonds are what produce our secondary structures. Big example of this is another way that they'll ask me questions, is where you see this. Our DNA double helix is an example of our secondary type structure. Secondary type structures are DNA double helix. Here we go. Here we get our alpha helix type structure or our beta pleated sheets. Now, tertiary, all of these different alpha helices and these beta pleated sheets, they also interact with each other. Overall, one of these alpha helixes might be slightly negative on one part or slightly positive on another. The beta pleated sheet might have parts that are positive, parts that are negative. These are also going to interact with each other and continue to fold. And that's how we get our tertiary structure. We get folding of a single polypeptide chain in a solution, and this actually can be a finished protein. We fold all of these sheets and everything together, and they can be a functional protein. Here is also typically where we enter in and we talk about disulfide bonds. Don't get me wrong, tertiary structure still has hydrogen bonding. It still has peptide bonding. It has those. But what makes it unique is this is where we see disulfide bonds happen. Okay, so if they ask you about protein structure and where you see disulfide bonds, it's in the tertiary structure. A good example of tertiary structure, where it is the final finished protein, is myoglobin. Break this down. Anytime that we see myo is referring to muscle. Globin, another one that we talk about similarly, we talk about hemoglobin. The globin referring to a globular type protein with this tertiary structure. This is our myoglobin. So, and the myoglobin's job is to carry oxygen inside of the muscle. Okay, this is another strategy when we talk about learning all this material. Take the words, break each of them down into their individual components. If you understand what each part of the word means, you can figure out what a word means that maybe you have never seen before, like myoglobin. Quaternary structure. This is a step beyond tertiary, where we take multiple tertiary structures and they all fit nicely in together to form a larger protein. Oftentimes, this is going to form a protein that has multiple functions, or it can do multiple things. So this is an association of two or more polypeptide chains to make a functional protein. Hemoglobin is the classic example. So myoglobin, for example, it has space and it carries one oxygen. Hemoglobin is 4 tertiary proteins. It's 4 polypeptide chains that all come together and in order to form a structure, a protein that can carry 4 molecules of oxygen. Hemoglobin is our best one for talking about quaternary type structure. Once again, we have our hydrogen bonds and disulfide bonds. And you can also see here another type of interaction called van der Waals forces. These are the 3 types of interactions / bonds that occur within a quaternary structure. So here we see there's our tertiary structure like our myoglobin, and this is our hemoglobin, 4 of those put together. All this of protein synthesis is going to take place in these structures known as ribosomes. We think back to our cellular biology. Where are ribosomes located? Ribosomes are located in our rough endoplasmic reticulum. So remember, we have the nucleus where DNA is held in the nucleus. We scan that DNA. We create a messenger RNA. We ship that out. It goes to the rough endoplasmic reticulum. It gets read. It gets processed within the ribosomes. That produces our proteins, and then we have something called a Golgi complex, which is responsible for packing and moving all of those things. I'm not sure if that will come back up. I'm not 100% sure that will come back up later. It's not usually asked in that kind of detail in part 1, just because it is more of a basic topic. However, if we do not see it in future videos, I will try and make a small refresher on our cellular biology. Okay, and that is going to end it for this video. We're going to stick with that with our protein structure.